Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich, and that is not me. What that is supposed to be representing is kind of a morph between modern Homo sapiens and Homo sapiens neanderthalensis, and some sort of maybe hybrid in between the two. I don't know, whatever. In this notes cast, we're going to be talking about evolution. We're introducing evolution as a concept and how it is something that does actually take place, and we do have evidence for that. Evolution. A lot of people associate evolution with Charles Darwin, and here's good old Chucky D in 1854 looking rather severe. And some people think that he's the first person to define evolution and talk about things changing over time. He's not. He was a big fan of Charles Lyell, a geologist who spent his career discussing how the Earth has changed over time. And so geological evolution is what you guys all studied, most of you anyway, last year in Earth Science. Makes sense then that organisms are also going to change over time as the Earth changes over time. Different ones will be better suited for the environments that come and will do better as areas change. And Charles Darwin's claim to fame is to have described a mechanism by which this takes place, natural selection, which we'll talk about later on. In this video, we're just going to focus on the evidence that this change has taken place. One of the places that we can look for this evidence is in the fossil record. Most of the time when we're talking about fossils, we're talking about extinct organisms, those things that are no longer found anywhere alive, like good old Mastodon giganteus here. The opposite of extinct is extant. This is where living specimens are still found. Kind of in between these two is like the mountain lion here in New York State, which is considered extirpated, which means that they're still around on the planet, just not in this area, even though they used to be in this area. Of course, this is controversial. According to DC, there are no mountain lions in New York State. There haven't been any carcasses found, at least at this time. There haven't been any carcasses found or poops or any other sign that's been officially confirmed. But like I said, a lot of people think otherwise. So we should talk a little bit about the three different types of fossils. Download the notes sheet if you haven't already. It's on MrUlrichsLandofBiology.com. If you didn't get there from my website, makes it easier to take some notes. Okay, one of the types of fossils are, well, it's really two, but we really lump these two together. Molds and casts. This is where something dies or makes a dent somehow by walking, like that rather large T-Rex there on the left. It makes a dent in the mud, and the mud solidifies into stone. Oftentimes, the inside of the dent will get filled in, as it did over here with Rhamphorhynchus. Those aren't the actual bones. There's no bones left in this here. What that is the dent that was then filled in with another material, making the cast. Another type of fossil is petrification. Again, this is, though that looks like a tree, there's no part of a tree left. It has literally turned to stone. That's what petra or petrus means, stone, rock. And what happens is the tree dies, falls over, gets covered over in sediment, and as water moves slowly through the sediment, it will slowly wash away, erode, degrade the carbon molecules, carbon atoms really, and all of the molecules that they're associated with. And we'll wash those away and replace them with dissolved minerals in the water. So you get kind of a mineral by mineral replacement of the tree molecules, if I can be so general, but replacing the individual molecules with minerals. And so you get this kind of mineral facsimile of the original tree. You can even count the rings. Pretty cool stuff. Last type of fossil, or group of fossils that we'll talk about, are when the organism itself, and in this case, there actually are remnants of the organism left behind. These are stuck in amber, frozen in ice, or somehow make it into peat bogs. Again, these do contain actual tissues from the original organism so they can be analyzed, which is pretty cool. Fossils are certainly cool, but we're interested in change over time, so we need to be able to put these fossils in order and see how they've changed over the millennia. So relative dating is a way of knowing if something is older than or younger than something else. It doesn't give us the absolute date, which we'll talk about here in a second. One way of doing this, the same method that I use for organizing my desk and your lab folders for that matter, and that's the law of superposition. And this it states that those fossils that are found in the upper layers are younger than those fossils that are found in the lower layers. Now, the nice thing, of course, is that the layers don't need to be directly above one another. They can also be located further away, and we can correlate these layers by looking for homologies in the structure of the soil as well as the fossils that are in them. So we can kind of get a timetable here of fossil, fossil, and we can see kind of similar. Here's that same fossil, here's that same fossil, here's that same fossil. And that way we know that this fossil here is much older than this fossil here, or even this fossil here, even though they could be miles away from one another. Even cooler, even more helpful would be if we knew exactly how old one of those fossils were. If that were the case, then that fossil would be an index fossil. So if this fossil here turned out to be 350 million years old, then we know that these are younger than 350 million years old, and I know that these are older than 350 million years old. So if we're going to have index fossils, then we have to have a way of determining exactly how old fossils are. And absolute dating is doing just that, finding out exactly how old something is. Radio dating is a mechanism to do this. Carbon-14 radio dating is real common to use with living things, of course. Carbon-14 is a naturally occurring isotope. It's out there in the environment. It's in you, it's in me, it's in the food that we eat. And as we live our lives and assimilate carbon from the environment, we kind of maintain the same concentration of carbon-14 in our tissues. But once an organism stops assimilating carbon from the environment, it's no longer bringing in that carbon-14. And since it does break down, and it breaks down at a known rate, if I can look at something and I find out how much carbon-14 is in it, and I know how quickly it breaks down, it's a simple matter of calculating how much time has elapsed since that organism stopped assimilating carbon. Fossils are neat and all, and I like them. But I'm also a biologist, so I like to look for evidence from living things. A lot of times when we're looking for evidence from living things, we're looking for genetic relationships. So we're looking for signs and signals of genetic relationships. Just like two people who are more closely related are going to have more genes and more structures in common. My older brother and I share quite a few genetic traits. Two individuals, two species that are more closely related are going to show more traits that are in common. One place we can look for this, we don't have to wait for them to be all grown up. We can look at them while they're developing. This is called comparative embryology. If we look at the chicken embryo over here on the left compared to the human embryo on the right, though chickens have a tail in their lifetime, certainly humans do not as grown-ups, and neither of us have gill pouches, but we do have these common structures, and since structures like this come from genes, this does show some degree of relatedness. We can just skip the structures altogether and look right at the genes these days. Uh, a kind of dated technology, but it still makes sense to discuss it. Electrophoresis is one technique that we can use to look at genetic similarities. But we can also look at similarities in proteins like hemoglobin, how close are the structures of the proteins. Again, since those proteins come from genes, we're really looking for genetic similarities when we're looking for protein similarities. We can also look at the anatomy, the structures themselves of organisms. Um, Sometimes these structures can be a little misleading because they can have wildly different functions, but still have similar enough structure that there are genetic similarities. So remember, how an organism uses their structures is not necessarily genetic, certainly not as genetic as the way that that structure develops and what it's made of. When we do comparative anatomy, we're taking a look at structures in two different organisms that they may have different uses, may have similar origins, and they may be similarly structured on the inside. These types of structures that have wildly different uses. When we look at the wing of the hummingbird and the forefluke of the baleen whale, though they're wildly different in size, the internal structure and the way that those things develop is very similar. They both have a single upper arm bone, the humerus, two parallel bones, the radius and ulna, some carpals in there, and then some phalanges. And so, because of this, that does show that they share some genes in common, since they have such similar structures that are made from the same materials and therefore require the same genes. It doesn't matter that they're used for wildly different purposes, because what an organism uses the structure for is more or less not encoded in the genes. These homologous structures show strong evolutionary relationships. That's the language that's typically used, which means that the common ancestor has been more recent. The opposite of homologous structures are analogous structures. These are structures that, though they might look similar from the outside, both butterfly and bird wings are flat, flappy things that they use for flying. The internal structure of them is wildly different. Birds have an endoskeleton made of calciferous bone, where the butterflies have an exoskeleton made of chitin. So even though they have that similar external appearance and they're used for the same thing, the internal structure is totally different, and that means, since they're different materials, that means that they come from totally different genes. This shows a weak evolutionary relationship. So there may be an evolutionary relationship. There are other homologies between butterflies and birds. They're both eukaryotes. They're both multicellular. They both use collagen to glue their cells together. They're both heterotrophs. So there are some homologies between the two, but they don't show a very recent common ancestor. The last type of structure we'll talk about is really another category of homologous structures. These are called vestigial structures. That little thing back there are the hips of the whale. We don't usually think of whales as having hips or having hind limbs, because they don't have hind limbs. But these are a type of homologous structure. They are remnants of hips, and that's what a vestige is. We talk about a vestige of something that is a leftover or a remnant. So in this case, our hips have a use, supporting our weight while we walk and giving leverage for our legs to move. Whales certainly don't use their hips for anything. They are reduced in function to functionless. These do show a strong evolutionary relationship, a recent ancestor. The thing that you want to avoid, the trap that you want to avoid falling into, is this idea that what makes a vestigial structure vestigial is the fact that it doesn't have a use anymore. Got to be careful. You can do very well without a pinky. You can chop that thing right off, and you're going to do okay. You'd probably rather keep your pinky, but you'd still be able to survive. So that doesn't make your pinky a vestigial structure. Your appendix, however, is a vestigial structure. That's a little sac that used to be a place for bacteria to hang out for you to digest your food, but it became more of a cost to have a blind pouch filled with food and bacteria in your digestive tract than a benefit of having all those bacteria to help you digest your food. But we'll talk more about that later on. We get into natural selection. At this point, just recognize that the vestigial structures are types of homologous structures where the use is so different that one no longer has that use. Well, we'll stop there for now. Again, thanks for checking in. And I'm Mr. Ulrich. And please make sure if you have any questions or comments or any other things that you want to talk to me about, come over to MrUlrichLandofBiology.com. Drop me a line there at the email that's coming up, and we'll see you in class.